What's up, biology students? Mr. Holloway here. In today's video, we're going to talk about what happens when the genetic code of life is altered, when our DNA is mutated. You probably won't become a superhero, unfortunately, but it is still important to understand, especially since mutations can result from things we expose ourselves to every day and can, in some cases, make our lives significantly worse. In our last video, we talked a bit about gene expression. Remember, when a gene is being expressed, that means that that gene is active and is being used to make proteins. But what happens if we cause the sequence of that gene to change? What effect could this have? Our genes hold information that the cells can use to assemble protein molecules. According to the central dogma, genetic information in the cell flows from DNA to RNA to proteins. And these proteins perform a huge variety of functions inside of our cells, functions which ultimately give us our traits and our abilities. Essentially, this diagram here is showing us what happens when a gene is being expressed by the cell. If you understand how this process works, it should be fairly obvious that changing our DNA means changing the proteins that our DNA codes for. But there are many different ways that this could play out, sometimes beneficially, sometimes harmfully, and fortunately for us, most of the time with no effect whatsoever. A mutation is any permanent change that occurs to the sequence of nitrogenous bases in our DNA. And there are basically two kinds of mutations that could ultimately affect the proteins our cells are able to manufacture. The first of these is called a point mutation, which is a mutation that occurs in only one point or only one location by swapping out one nitrogenous base for another. Here at the top of this slide is a normal, non-mutated sequence of DNA that we will be using for reference during this lesson. The blue strand on top represents the template or antisense strand of DNA. Based on this template, a strand of mRNA containing 4 codons can be synthesized. These 4 codons instruct the cell to build a polypeptide with 4 amino acids, in this case, phenylalanine, tyrosine, alanine, and arginine. In our first example, a point mutation has occurred at this location, and the A base that was supposed to be there was replaced with a G base. As a result, the mRNA synthesized from this DNA contains a C at this location instead of a U. However, if we look up the codon UUC in our codon chart, the same chart we saw in a previous video, we will find that both the original UUU codon and the mutated UUC codon both code for the amino acid phenylalanine. This is called a silent mutation because the mutation has no effect on the protein being assembled, since the same sequence of amino acids is produced in both cases. If you look back at this color coding chart, you should notice that many amino acids are coded for by more than one codon. And in almost all cases, it is the third base in a codon that varies between codons coding for the same amino acid, just as both UUU and UUC coded for phenylalanine in our first example here. Let's look at another example, another point mutation called a missense mutation, but at a different location. This time, our DNA contains a C at this location when it should contain a G. This means that instead of the GCA codon we were supposed to have, we have the codon GGA embedded in our strand of mRNA. The original GCA codon corresponds to the amino acid alanine, but the codon GGA corresponds to the amino acid glycine. The end result is a slightly different sequence of amino acids and a slightly different protein molecule as a result. This difference in amino acids may or may not affect how the final protein functions. The final protein may work just as it was supposed to, or its function may be altered, possibly for better or possibly for worse. But since this mutation is a permanent change in the DNA in this cell, any proteins built from this gene will all contain this alteration. But suppose a point mutation results in a misplaced start or stop codon, and not just a different amino acid. This kind of point mutation is called a nonsense mutation, and we can see an example of that here. In this case, a point mutation at this location has resulted in a T base where there was supposed to be an A base. When our DNA is transcribed to make RNA, this change in our DNA results in the codon UAA being transcribed instead of the codon UAU, which is what we were supposed to have. UAU codes for the amino acid tyrosine, which was supposed to be the second amino acid in our polypeptide chain. But the codon UAA is a stop codon, and when a ribosome reaches this codon, it will stop building the protein, and our amino acid chain will be much shorter than it was supposed to be. Depending on where in the gene this kind of mutation occurs, we could end up with a mostly complete protein or one that contains way fewer amino acids than it is supposed to. But in either case, the overall function of that protein is very likely to be altered, and it's likely that that protein won't function the way it was supposed to. Some genetic mutations are called frameshift mutations instead of point mutations, and these occur when a base is deleted or inserted into our DNA. Frameshift mutations result in a completely different set of codons being transcribed into our RNA. Let's take a look at how this works. In this first example, we're looking at an insertion, a frameshift mutation in which an extra base is inserted into the genetic code, but no bases are removed to compensate. In this case, we've got an extra T base inserted into our DNA at this location. Notice how all of the bases that follow this extra T are the same as they were when we started, just moved one position to the right. This is why we call it a frame shift, because everything shifts one way or the other depending on whether you add an extra base or take away a base that was needed. This means that every codon that follows this mutation is messed up, since the last base in this codon is now the first base in the next codon as a result of this mutation. The codon UAU is now AUA. The next codon, which was supposed to be GCA, is now UGC. The last codon, which was supposed to be GCU, is now ACG. Every base got moved down one space, but the so-called reading frames around our codon stayed the same. As a result, we've built a completely different amino acid sequence, and a different protein than the one we were supposed to build. This protein will not function the way it was supposed to function, and this could have serious consequences for the organism with this mutation. If, for example, this protein was supposed to help the cell metabolize a particular molecule or transport a particular substance across the cell membrane. There's also a small chance that this kind of mutation could be beneficial to the organism involved, allowing it to digest a new food source or giving it a new immunity to some kind of pathogen. And these kinds of beneficial mutations are a huge driving force in the process of evolution, but we'll learn about that process a little bit later in the year. Likewise, if we simply remove a base from our gene, all the remaining bases will shift one spot to the left, another example of a frame shift mutation. In this case, we've removed a T base from this location. Once again, this causes all the codons that come after this mutation to shift. The codon that was supposed to read UAU is now UUG. The codon that was supposed to read GCA now reads CAC, and so forth. Just like our last example, this means that our entire amino acid sequence will be altered, and the protein we are building will be fundamentally different than the one we were trying to build. Its function will be different as well, and it may in fact not function at all, and this could potentially have very serious consequences for the organism involved. While some genetic mutations result in negative consequences, others may result in positive, beneficial changes. But the vast majority of the time, more often than not, when a mutation occurs, it has no effect whatsoever, which is a pretty good thing for us actually. Think back to our first example, the silent mutation. Mutations like these do not change the sequence of amino acids coded for by a gene, and as a result, have no effect on the protein manufactured from this gene. Now think back even farther. Remember when we were talking about transcription and translation, and we talked about how our DNA contains these regions called introns that do not code for anything and which are edited out of our RNA before this RNA leaves the nucleus? That process is depicted again here. These introns make up a surprisingly large portion of our DNA, over 90% according to some estimates. If a mutation occurs in these regions, in these introns, even if it's a serious mutation that results in big changes to the genetic sequence, it will have no effect whatsoever because these introns do not code for anything and will in no way affect the kinds of proteins we are able to make in our cells because these sequences are edited out of our RNA before it leaves the nucleus. Think of it as a built-in defense mechanism, and a good one considering how many mutagenic agents we expose ourselves to every day. Because we know that mutations have the potential to be harmful, even if most of them are not, it is useful for us to know what kinds of things cause mutations in our DNA. These things that cause mutations are called mutagens, or mutagenic agents, and we expose ourselves to some of these almost every day. Poisons and toxins in our environment can cause mutations, things like formaldehyde, benzene, and arsenic. Many of the ingredients and materials found in cigarettes, chew, and snuff are mutagenic and are known to greatly increase the risk of developing mutations that lead to lung, throat, and mouth cancers, which is why the Surgeon General puts a warning on all packs of cigarettes and all tobacco products, warning people that the materials inside of these substances are known to potentially cause cancer. And they're not joking, it does happen. Radiation and radioactivity are known mutagenic agents as well, and can sometimes be difficult to avoid. The sun, for example, is an enormous source of radiation in our daily lives, and most of us are exposed to radiation from the sun way more than from any other source. Exposure to the sun does a lot of good things for us as well, like helping our bodies to synthesize vitamin D, but too much sun exposure can be bad for us. That's why it's important to wear sunscreen or protective clothing if you plan to be out in the sun for prolonged periods of time, so as to limit your exposure to potentially mutagenic radiation. But radiation can come from other sources too. Radon gas seeping into your basement is radioactive and can expose us to small amounts of radiation, especially when allowed to build up, as often occurs during the winter when folks have their homes sealed against the cold weather outside. In the doctor's office, things like X-rays and CT scans expose us to additional radiation as well. Even our cell phones, TVs, and computers expose us to radiation. However, it's also important to note that when it comes to radiation, the effects are cumulative, and the tiny amounts of radiation we are exposed to from cell phones and computers pales in comparison to the amount of radiation we receive from the sun every year. And because most of the mutations that occur in our DNA are completely harmless, it takes a lot of radiation to increase our risks of developing mutations that can lead to things like cancer. For example, you would have to have approximately 1,000 chest x-rays before your risk of developing cancer even registered as an increase on the scale. And x-rays put out way more radiation than any cell phone on the market. By comparison, simply living at sea level for 50 years or living in Colorado for 20 years would expose you to the same amount of radiation from the sun as you would be exposed to from those 1,000 chest x-rays. On the other hand, 10 full-body CT scans, or approximately 11 years working as an airline pilot, would also expose you to the same amount of radiation as 1,000 chest x-rays. One study revealed that regular use of tanning beds, once a month or more for almost a lifetime, increases a person's risk of developing skin cancer by as much as 55%. So not all radiation is insignificant, I guess, but remember, it's not like a little radiation is going to kill you, and you're actually exposed to a little bit of radiation in pretty much anything you do on any given day of the week. The point is, since we know that the effects of radiation are cumulative, it's important to try to limit your exposure when you can. Not that a day in the sun is going to kill anyone, but long-term exposure to radiation can increase your risk of developing harmful mutations that could result in things like cancer, disease, sterility, and a variety of other serious health problems. So, no one's going to get cancer from using a cell phone. That's the good news. And it's also good news that the vast majority of mutations that occur in our DNA are completely harmless and benign. But some mutations do alter the structure and therefore the function of proteins manufactured by our cells. And the consequences of these alterations is just as likely to be very bad for us as it is to be even a little bit good for us. So protect your DNA and try not to overexpose yourself to anything that might cause your genetic code to mutate. And with that, I will bring our video to a close. Thanks for watching, and feel free to go back and watch this video as many times as you need until you feel like you understand how mutations in our DNA affect us and the proteins manufactured by our cells.